Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Ditoro Magwala, and this lesson is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So in today's lesson, we are looking at our paper 2 final exam preparation. So here we are just going to be getting a reminder of all those things that we must have a look at before we go into our final exam, right? So just a reminder, our paper is 2.5 hours long and it is worth 150 marks. So it is 150 minutes for 150 marks, meaning that you must make every minute count. All right. So our paper is a 2-section paper, section A and section B. And in section A, we have short questions, which are worth 50 marks, right? So we have your multiple choice questions. We have your terminologies or biological terms. We have the matching column, as well as a few short questions. All right. And then in section B, we have question 2 and question 3. Question 2 and question 3 both have long questions. So here you're going to be finding your drawing questions, your graphs, your investigation questions, right? Each question is worth 50 marks, and that would be a total of 100 marks, and then the grand total for your paper will be 150 marks. Now, what topics do we have in paper 2, right? Here we have DNA, the code of life, which is 27 marks. We have meiosis, which is 21 marks. That is the shortest part. We also have genetics and inheritance for 48 marks. And lastly, evolution, which is our longest, for 54 marks, right? So we are just going to be looking at each topic and looking at the main things that you should have a look at in order to make sure that you are ready for your exam, right? Before we get into that, let's have a look at our marking guidelines and tips. Remember, when we look at the marking principles, we're just having a look at this because these are the principles that are used by your markers, right? Meaning that as they mark, they are going to be following these principles. Therefore, this will help you to know exactly what they will be looking for, and that means those are the things that you should have in your paper to make sure that you pass, right? The first one, if more information than marks allocated is given, stop marking when maximum marks are reached and place a wavy line and max on the right hand margin, meaning that you do not need to give too much information for little marks. Make sure that you give enough information for the marks that are allocated. Okay, if whole process is given when only part of it is required, read all and credit the relevant part. So if you do not quite know which part the question is asking for, you can give a whole process, and from that whole process, the required part will be marked correct and you will not lose any marks. If comparisons are asked for and descriptions are given, accept if differences and similarities are clear, right? So here, if you are asked to give similarities or differences and you are giving a description, your description must at least contain those differences and those similarities in order for you to get the marks clearly stated, right? And then with your differences, you would usually be asked to give differences in the form of a table. They will say tabulate the differences, meaning that if you give this in the form of a description, you're then going to lose that 1 mark for tabulation. Remember, we're not trying to be losing any marks here. Okay, so make sure that if the question says tabulate, you are going to give your answer in the form of a table, whereby you are going to indicate that this is, let's say the question says differentiate between DNA and RNA. On one side, you would indicate that this is the difference of the DNA and this is for RNA, so that it can clearly state that. And then you're then going to get a mark for tabulation. So you would draw a table that looks like this, and you would say DNA and RNA, right? Remember, every drawing must have a caption, right? If flowcharts and diagrams are given with annotations when descriptions are required, candidates will lose marks. We do not give flowcharts, we do not give diagrams when the question is asking for a description. You are going to describe in words, right? Because you would then lose marks. It is not going to be marked at all. So imagine if the question is asking for a description of transcription, right, for 6 marks, and you draw transcription and you show that this is the nucleus, this is DNA, this is RNA leaving the nucleus, and so on, you're then going to lose a mark because you have given a diagram with annotations instead of describing the process in words. So make sure that you describe. Captions: all illustrations, which are diagrams, graphs, as well as tables, must have a caption. If you draw, make sure you have a caption. If you draw a graph, it must have a caption. When you draw graphs, make sure that your graph has both variables in the caption, right? The dependent variable and the independent variable are mentioned in the caption. If that is not the case, then you will not be getting that mark for your caption. So make sure you write the correct caption. Okay. If only a letter is asked for, but only the name is given and vice versa, candidates will lose a mark. Now, here, if it says give the letter, you give the letter. If you don't know the letter, but you know the name, you will not get the mark, right? So it goes back to learning your structures, knowing how to identify them, and knowing how to name them. Okay. And then if you give the letter instead of the name, again, you are going to lose those marks. And you must be careful. Some questions would say give the letter and name, meaning that this would be a 2-mark question. Make sure you give both so that you do not lose those marks. All right. The next one, it says read all the information provided before answering questions. Make sure you read this information so that you know exactly what you are going to be answering. You know exactly what the question is all about before you even get to the questions. Because if you rush to answering the questions without understanding, then you would lose marks as well. Okay. For example, when you are drawing a graph, or when you're looking at an investigation question, you would find that the information there tells you what the independent variable and what the dependent variable is. Now, if you have not read that, you would lose marks on that. So make sure that you read so that you get the relevant information from that small paragraph that you have at the beginning of your question. Okay. Lastly, know all your terminologies. We have section A, which has a lot of questions that are based on your knowledge of terminologies. Now, for every topic or for every chapter that you're going to go through, make sure that you have those terminologies. Now, if we look at a chapter such as genetics and inheritance, there's so many terminologies there that can actually be a bit confusing if you do not understand them, right? So make sure that you know those so that as you come across these questions, you can easily understand what the question is all about and you know how to answer that question. All right, now let's have a look at DNA, starting with the structure of a nucleotide, right? So remember, a nucleotide is made up of 3 parts, right? Now, what are we talking about when we're talking about a nucleotide? We're talking about the building block of nucleic acids. We are looking at the building blocks of DNA and RNA. So this nucleotide is a building block of DNA and RNA, right? So each nucleotide or each building block is made up of 3 parts: a phosphate, which we see here, a nitrogenous base, which is what we are seeing here, as well as a sugar, right? So you should be able to draw and label this diagram of a nucleotide, right? You should be able to say, this is a phosphate, this is a sugar, and this is a nitrogenous base. And you should always remember that the phosphate is always attached or bonded to the sugar, and the sugar is always bonded to the nitrogenous base. Now, if you come across a nucleotide whereby the phosphate is attached to the nitrogenous base, that is incorrect. So make sure that you know the sequence of how these 3 parts are bonded to each other. Now, as I've said, both DNA and RNA are nucleic acids and they are made up of the building blocks, nucleotides, right? Now, they have some differences that you should also know, right? The first one is that DNA only occurs in the nucleus, whereas RNA occurs in both the nucleus and the cytoplasm. Remember, RNA is formed in the nucleus during transcription, and then it moves out of the nucleus into the cytoplasm. Hence, we say it is also found in the cytoplasm. DNA is double-stranded. As we can see here, this is the double-stranded structure of a DNA molecule and is referred to as a double helix, whereas RNA is single-stranded and it is not a double helix. All right, so DNA is coiled and it is a double helix, whereas RNA is not coiled. Right, number 4, the long strands of DNA that we have. So DNA has long strands, whereas RNA has shorter strands. Okay, and then next we have the fact that the nitrogenous bases do differ in DNA and in RNA. In DNA, we have the nitrogenous base thymine, and in RNA, we have the nitrogenous base uracil. So it means that when you come across a nucleic acid and you see that the nitrogenous base there is uracil, then you know that this is an RNA molecule and not a DNA molecule. If you come across a DNA molecule and it has the nitrogenous base thymine, then you should also know that this is a DNA molecule and not an RNA molecule. All right. Now, we're moving on to DNA replication and why this process is important. What is the importance or the significance of DNA replication? Why is it important for this process of DNA replication to take place? Now, firstly, what is DNA replication? Remember, this is a process whereby the DNA molecule is going to make an exact copy of itself, right? So the DNA molecule makes a copy of itself, meaning that at the end of the process, we are going to have 2 DNA molecules that are the same. Why is that important? Okay, so this is important in both mitosis and meiosis, and mainly in mitosis, we look at the fact that we want those daughter cells that result from mitosis to have identical genetic material. Remember, during mitosis, we have 2 cells or 2 daughter cells that are genetically identical to each other and to the parent cell, meaning that their genetic material is going to be exactly the same. Now, when we're talking about this genetic material, we're talking about the DNA. So the DNA must be the same, which means that DNA replication is important for exactly that. DNA replication is important for making sure that at the end of mitosis, the 2 resulting cells have identical DNA or have identical genetic material, right? So they must contain identical genetic material, which is DNA, and this is the DNA that was present in the parent cell, right? Now, also here, we should know that during mitosis, the cells remain diploid and the number of chromosomes does not change. So the number of chromosomes remains the same as the original cell, and the 2 cells that have resulted will also have the same genetic material. Right. Okay. Now, during DNA replication, we are also going to ensure that these daughter cells contain the same amount of DNA, which is the number of chromosomes, right? And during meiosis, we know that 1 round of DNA replication is going to be followed by 2 rounds of the cell division. We have meiosis 1 and meiosis 2, unlike in mitosis, whereby we only have mitosis, right? So this is going to result in 4 haploid cells. The cells that are going to result here are going to have half the number of chromosomes, right? So this also makes sure that the number of chromosomes that are going to be shared amongst these cells is going to be half, right? So it just makes sure that there is enough genetic material for those cells to share at the end of meiosis. Okay, now we are going to look at the process of transcription, which is similar to DNA replication. So as we look at transcription, I'm just going to give you a reminder of how transcription differs with DNA replication so that you are able to describe both processes. You're able to describe the process of DNA replication, and you're also able to describe transcription, should one of these 2 be asked in the examination. Okay, next we have DNA profiling. First thing here, be able to differentiate between DNA profiling and a DNA profile. What is the difference between these 2? Very important. A DNA profile are the black bars that are left behind on X-ray film when an extract of DNA undergoes biochemical processes, right? So DNA is extracted from an individual and it undergoes biochemical processes to leave these black bars on an X-ray film in order to identify certain individuals based on their DNA material, right? Now, what is DNA profiling? This is the process of identifying an individual by comparing their DNA with that of another known DNA. So now, when we look at DNA profiling, we are looking at the technique. And when we are looking at a DNA profile, we are looking at this actual profile whereby you can see the profile of an individual, right? So be able to differentiate between the two. And the question is asking about a technique. The technique is DNA profiling. If the question is asking about a DNA profile, then you should also know what is a DNA profile as opposed to profiling, right? You will not get a mark if you mix these two. All right. So that process is done by comparing the bands on a DNA profile of an individual to those of a profile from a crime scene or another individual. So we can look at these bands of DNA that we are seeing here and compare them to each other just to see if they belong to the same individual or someone else. And this is what we use to identify different individuals. Now, we can use this to identify parents, we can use this to identify relatives, we can use this to identify offenders in a crime scene, right? So we are then going to be looking at that. So for example, here, this is a paternity testing example whereby DNA profiling is used, right? So we can use this to identify which parent belongs to the child, or which of these possible fathers is the father to this child, right? So we will compare the DNA profile of the mother to the child and the DNA profiles of these possible fathers, and then whoever has the bands that match with the father with the child then would be the father. We can also do this to find the person or the suspect who has committed this crime in a crime scene. So here we would be looking at a rape case whereby we have semen from the crime scene, we have the victim's DNA, and we have the DNA from all the suspects. Now, because your genetic material is the same throughout the body, here we have DNA from semen, right? And then for the suspect and the victim, the DNA that is used here might be a blood sample. As you can see, it's not semen, but it is a blood sample. It can be saliva, it can be hair, it can be any cells from your body, and it will still show the same results, or it will still show that the DNA belongs to that person. So how we identify these can differ. How we use DNA profiling in paternity testing and how we use it in a crime scene might differ, in the sense that here, when we are looking at paternity testing, we first have to compare the DNA profile of the child to the mother, and then the remaining bands that did not match with the mother on the child's profile should then match the father. If they do not all match the father, it means that that individual is not the father. Meaning that now, here we're going to look at a comparison of the profiles of the child from number 1 to number 6 with the profiles of the mother. Half of the bands must match with the mother and half of the bands must match with the father, because any individual or any child has genetic material 50% of the mother and 50% of the father. So band number 1, we can see it matches with that of the mother. So we can put a small tick there to show that it matches with the mother. Band number 2 does not match with the mother. We expect band number 2 to then match with one of these males. Band number 3 matches with the mother. Band number 4 does not match. Number 5 matches with the mother. Number 6 does not match. So now 3 out of 6 bands match with the mother, which is how it should be. The remaining 3 must match with the fathers or one father. Okay, number 2, already, because band number 2 does not match with individual A, we are already excluding him as the possible father, because all these remaining bands 2, 4, and 6, they must all match the father. So because they are not matching, we are excluding A, we're also excluding C, meaning that we can now only compare A, B, and D. Okay, now let's have a look at that. Number 4. Band number 4 matches with B. and it does not match with D. So we are then also excluding D. But to be sure, we're going to check 6. 6 is also matching with B. So we can conclude that the father of this child is individual number B. Now, with this one, because it's not 50-50, we do not expect 50% to be coming from somewhere and 50% to be coming from somewhere. We can match and see which of these bands on the semen mostly match with the bands of the suspect. And then from there, we can say, okay, suspect number 1 is definitely the one who committed the crime, or we can say none of them have actually committed the crime. Now, let's just have a look at that quickly. Okay, so if we check, the first one matches with the victim and suspect 3. The second one matches suspect 3 only. The third one, 3 again. So from here, we can already see that suspect number 3 is the one that committed the crime. Right, okay. Now let's have a look at protein synthesis. Here, remember, we look at transcription and translation. Right, these are the two phases or the two parts that make up the process of protein synthesis, whereby we are going to be looking at the production or manufacturing of proteins. Right, now starting with transcription, which is the first part that takes place in the nucleus. Okay, so what happens here? Firstly, the DNA double helix unwinds. As the DNA double helix unwinds, the weak hydrogen bonds between the nitrogenous bases are going to break. And as they unbreak, they are going to unzip or separate, right? So the two DNA strands are going to unzip. Then one of those DNA strands, now we have two DNA strands, one of those DNA strands are going to act as a template to build the mRNA. So we can see here they have separated, and only this one is acting as a template, whereas this one is not acting as a template. One of them is acting as a template in order for the mRNA to be built. Here we can see the mRNA, right, using the free RNA nucleotides from the nucleoplasm, which will then attach to the DNA. So RNA nucleotides are going to come in and pair with the DNA nitrogenous bases to form that chain of an mRNA molecule, right? Remember, during a complementary base pairing, we are going to see that cytosine is going to pair with guanine, whereas adenine will pair with uracil. Adenine pairs with uracil, or uracil pairs with adenine, because we are forming mRNA. We do not expect to see thymine being one of the nitrogenous bases on the nucleotides, that on the RNA nucleotides, right? So you should remember that there is no RNA, there's no thymine in RNA, right? Okay, now the mRNA, let's go back to transcription. Remember I said that we're going to look at transcription and compare it to DNA replication. So the first step of DNA replication is exactly the same as that of transcription. The second one also, the DNA will unzip and weak hydrogen bonds will break, is also the same. However, when we look at DNA replication, instead of saying one strand acts as a template, we are then going to say each strand acts as a template to build DNA. Remember, in DNA replication, we're building DNA using the free DNA nucleotides from the nucleoplasm. And then in that case, adenine will pair with thymine because it is DNA forming another DNA. In DNA replication, both the strands serve as templates, whereas in transcription, only one strand serves as a template. Okay, and then during translation, which takes place in the cytoplasm, the mRNA would have then left the nucleus, will leave the nucleus through the nucleopore into the cytoplasm, and the tRNA will pick up amino acids and complement the mRNA on the ribosome. So this tRNA here will bring in the relevant amino acids based on the codons on the mRNA that was formed in the nucleus. So based on that, tRNA will bring in the relevant amino acid, and those amino acids will combine with each other and form a long chain. So the anticodons on tRNA match the codons on the mRNA, right? And based on the anticodons on the tRNA, they are going to code for a specific amino acid, right? The amino acids become attached to each other by peptide bonds to form a peptide chain, and that peptide chain would then become a protein, and that is how we look at the process whereby proteins are formed, right? And then we are going to look at the fact that these codons or these anticodons can actually code for the same amino acid, meaning that we can find that, for example, ACA codes for 2 different amino acids, right? Meaning that you can see ACA and it will bring in a certain amino acid, and you can still see ACA again, but that ACA would then bring in a different amino acid that is different from the previous one. So you would find that you have ACA, but the amino acid is now different. As we can see, I have color-coded them, but they would still be bringing in the same amino acid. So you should remember that the amino acids might be different, but the codon or the anticodon would still then be the same. All right, let's have a look at an activity on protein synthesis, right? Okay, it says here a mutation has occurred on a section of an mRNA molecule as shown below. We have the original sequence and we have the mutated sequence. It means that on the mutated sequence, we are expecting to see some change. Remember, when you're looking at the mutation, we're looking at a change that has occurred. Right now, let's find out what change has happened. So we can see here it's still the same, but here we have CCG and CUG. Everything else is still the same. So this is where the mutation has happened. What kind of mutation has happened here? The C has been replaced by the U. It means that there has been a mutation over there. Okay. 2.2.1, it says the table below shows some mRNA codons and the amino acids that they code for. We have the mRNA codons and we have the amino acids. Like I said earlier, read the information provided. If you jump onto this part here, but you have not read the information, you might miss out on the fact that here we are looking at mRNA codons coding for amino acids. Remember, in another question, it might say tRNA anticodons, or it might say DNA triplets. Remember, in DNA, we have triplets when we are looking at the 3 nitrogenous base. In mRNA, we have codons, and in tRNA, we have anticodons. All right, so now let's have a look at the amino acids and the codons. Remember, these are mRNA codons. We can see here already we have proline twice. However, when we look at the codons, they are different. So proline can be coded for by 2 different codons. So if you come across a CCA, it can be proline. If you come across a CCG, it can still be a proline. Meaning that finding different codons on a DNA sequence does not necessarily mean that the amino acid will change. You must find out first. You must check, has this led to a change in the amino acid, or does it still code for the same amino acid? If it codes for the amino acid, it means that should any change happen or should any mutation happen, that mutation will not necessarily affect the amino acid that is going to be coded for, therefore does not change the protein formed. However, if it changes the amino acid, then the protein formed will also be different. Now let's jump into the questions and see. Okay, the first one says, state the number of different amino acids coded for by the original sequence of the mRNA molecule given above. Make sure you read your questions. Read that question. It says original sequence, meaning that we're going to look for this on the original sequence. Another thing that you must check, the number of different amino acids, not the number of amino acids. Because if you look for the number of amino acids, you would say 1, 2, 3, 4, 5, 6. That is the number of amino acids, right? But the question says the number of different amino acids, meaning that if you come across a codon that codes for the same amino acid 3 times, then you count it once because it's not different amino acids, but it's an amino acid. So now let's check how many different amino acids do we have here. So we have AUG, we have GAA. What does GAA code for? It codes for glutamic acid. We have AUA. Now, because here we're looking at an mRNA and we're also looking at an mRNA table, it means that we do not move from mRNA to tRNA to make our comparison. It is the same, right? The next one was AUA. We have AUA, which is isoleucine, right? And then CCG. What does CCG give us? CCG gives us proline, right? Proline here. And then glutamic acid here. And then what does it give us here? CCA. CCA gives us proline again, meaning that we have proline twice. GGA, what does GGA give us? GGA gives us glycine. We have not come across glycine. So it means that this is glutamic acid, this is glycine. They are not the same. So what does AUG give us? AUG gives us methionine. Methionine here. GAA. GAA gives us glutamic acid. AUA, okay, this is glutamic acid. And this is isoleucine. Now, how many different amino acids do we have? 1, 2, 3, 4, 5. We do not have 6 different amino acids. We have 6 amino acids, but 5 different amino acids. So that would be our answer. B, give the anticodon on the tRNA molecule that carries the amino acid isoleucine. Right. So now, if we are looking at the anticodon for isoleucine, it means that we must first check the codon for isoleucine and then give the anticodon. The question wants the anticodon for isoleucine. What do you have? You have the codon for isoleucine. Right? Let's check the codon for isoleucine. AUA. So if we have AUA, what does AUA pair with? AUA would pair with UAU. Remember, we do not have T, we do not have thymine. So it will be UAU. That would be our anticodon for isoleucine. Right, there we have it. And then next, use the information in the table to describe the effect of the mutation on the protein formed. Right. Remember, there is a mutation. Right. What is the mutation? Let's have a look again. Instead of CCG, we have CUG. Right. Now, if we have CUG instead of CCG, what difference does it now make? Let's see what CUG codes for. CUG codes for leucine, right? Because CUG codes for leucine and CCG codes for proline, it means that the amino acid has changed. And if the amino acid has changed, it means that the protein formed is going to change as well. Right, so you're going to mention that there has been a change in the nitrogenous base CCG to CUG, whereby C has been replaced by U, right? And then the amino acid proline has been replaced by leucine, which then means that the protein formed is also going to change, right? So the codon CCG changed to CUG, right? The anticodon tRNA sequence has changed, right? So the tRNA sequence has changed, and the amino acid proline has now been replaced by leucine. This will result in a different protein, right? However, let's say, for example, that CCG was replaced by something that will give us the same amino acid. If the CCG was replaced by CCA, the amino acid formed will still be the same, therefore the protein formed will still be the same. And that is what you would have said. You mentioned that there has been a change in the nitrogenous bases and that there has been no change in the protein formed because the amino acid that those two codons code for is the same, right? So do not just always claim that a change in the nitrogenous base will lead to a change in the amino acid, because sometimes it does not lead to the change in an amino acid. Now let's have a look at question 2.2. It says, name and describe the process where tRNA plays a role in the formation of a protein. When we talk about tRNA, we are talking about translation. The question says, name and describe the process. It means that the first thing that you're going to say is translation, and then you're going to get a mark for that. This is a compulsory mark because the question said name and describe, meaning out of 5, 1 mark already is going towards the naming. 4 marks is for the description. If you describe without naming, you lose that 1 mark for naming. Okay, let's have a look at that. Each tRNA carries a specific amino acid. We know this. When the anticodon on the tRNA matches to the codon on the mRNA, then the tRNA will bring that required amino acid to the ribosome, and those amino acids will become attached to form a peptide chain. Right, now that is our protein synthesis, whereby we look at transcription and translation. Next, we have meiosis. Right, with meiosis, we first look at the karyotype. Right, so with the karyotype, remember, you should be able to differentiate between a male karyotype and a female karyotype. This is a female karyotype because we have XX chromosomes on our gonosomes, right? Our sex chromosomes are XX. On the male karyotype, you would identify this by seeing the XY. Right, how many pairs of chromosomes do we have? We have 23 pairs, right? From pair number 1 up to pair number 23 on both, as we can see there. The 22 pairs, these are referred to as autosomes, right? And then the last pair, they are referred to as the gonosomes or the sex chromosomes, right? These sex chromosomes are the ones that are responsible for determining the gender of an individual or the sex of an individual, right? So the 23 pairs, autosomes, 22 plus the gonosomes on pair number 23. In females, the gonosomes are XX. In males, the gonosomes are XY. All right. And then now let's have a look at the phases of meiosis, right? When we're looking at the phases of meiosis, we are just going to have an overview of both meiosis 1 and meiosis 2. Remember, before the beginning of meiosis, we are going to come across interphase. This is the phase that takes place in preparation for meiosis, right? It takes place before meiosis 1 can start. And remember, at the end of meiosis 1, before meiosis 2 starts, we do not have interphase. It only takes place once at the beginning, right? So you should be able to identify all these phases from interphase all the way until cytokinesis 2. When you look at a diagram, if you're provided with a diagram in the examination room, you should be able to identify that this is interphase, right? You should be able to say this is anaphase 2, this is telophase 1, by knowing exactly what happens during each phase. So you should know what happens during prophase 1, for example. You should know that here we have crossing over taking place, right? And the chromosome numbers are still double what we would come across at the end of meiosis, right? Then you should know what happens during metaphase. You should know that during metaphase, we're going to see the chromosomes aligning at the equator in homologous pairs, and they are aligning randomly. Anaphase, the chromosomes are going to separate. Whole chromosomes are going to be pulled to the opposite poles, right? Telophase, we are seeing now the division of the cytoplasm is taking place, and the chromosomes are now showing that they are in distinct cells, right? Cytokinesis, now we can see the two haploid cells. Remember, when the separation happens here, the chromosome number now is going to halve. Now we have N, and we do not have 2N anymore. Now, these two cells at the end of meiosis 1 are going to go into meiosis 2, and they are going to undergo meiosis 2, whereby now you must be able to identify how is prophase 1 different from prophase 2. In prophase 2, we do not have crossing over, right? That's how you differentiate between prophase 1 and prophase 2. Right. However, you would see that the centriole will now be forming the spindle fibers, and they will be moving to the opposite poles. You will see the disappearing of the nuclear membrane and the nucleolus taking place there. Metaphase 1. How does metaphase 2 differ from metaphase 1? In metaphase 2, the chromosomes are aligned singly at the equator, not in homologous pairs. Here you can see there's 2 chromosomes. Here there's 1 chromosome. Homologous pairs and singly. They align at the equator, and you would see that in anaphase 2, chromatids are now being pulled to the opposite poles. Here you can see the 2 chromatids are going to the same pole, right? Now, you must also keep in mind that whatever is happening in the cell will also be happening in the second cell. Okay, and then we have telophase 2, whereby we are going to see that now the chromatids are going to be forming part of the 2 new cells that are going to be formed. And at the end of cytokinesis 2, as we can see, we have 4 cells, and each cell is haploid. We started off with 1, 2, 3, 4 chromosomes. Now each cell has 2 chromosomes, and those chromosomes are unreplicated. Do not be confused by the fact that here we have what we know as chromatids. They are still referred to as chromosomes. They are referred to as unreplicated chromosomes. So now we have half the number of chromosomes. Now, by knowing that this is telophase 2, for example, and it has 2 chromosomes, it is a guide for you that if during telophase 2 we have 2 chromosomes, it means that during prophase 1, we had 4 chromosomes, because now during telophase 2, the chromosome numbers have been halved. You will be asked questions about how many chromosomes were at the beginning of the process, or how many chromosomes will be at the end of the process. Remember, chromosome numbers are halved during meiosis. If you start with 20, you will end up with 10. If you are ending up with 20, it means that you started off with 40. So at the beginning, it's double. In the end, it's half. Okay,